Hi, everyone. I've got a very quick and exciting announcement here. We're hosting our very first ever in person meetup on November 7th from 6 p.m. to 8 p.m. in Denver at the ASHG conference. It's going to be a fantastic opportunity for past guests, future guests, listeners, and friends of the show to come together, share ideas, and chat about all things genetics and precision medicine. We have reserved a very cool venue. We're going to have free food and drinks, and there's going to be a lot of interesting people in attendance. We're sharing this event first with listeners of the podcast as well as past guests. So if you can be there, please register as soon as possible because the spaces are limited. You can register at lu.ma/geneticspodASHG, all lowercase, no spaces or dashes in any of the words. The link to register is also in the show notes and it's easy to use on your phone. Again, that's lu.ma/geneticspodASHG, all lowercase. Mark your calendars, come say hi, and I can't wait to meet many of you in person. One of the challenges of podcasting is you often have absolutely no clue who the listeners are on the other end, except those of you who I really appreciate who drop me an email or say something on LinkedIn. But that's actually a very small minority of people. So if you can't make this meetup for any reason, but you really like the idea, then please don't hesitate to drop me an email at patrick@sonogenetics.com just to say hi. Let us know where you're based so we can keep it in mind for future meetups. And if people like this sort of thing, then we're going to plan to do more in the future. So thank you again, and we look forward to seeing you there. Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hi, everyone. Welcome back to the Genetics Podcast. I'm here today with Dr. Veera Rajagopal, who's a drug discovery scientist at Regeneron. If you haven't heard his name before, then this is probably one of your first episodes with the podcast, but we do this every 3 months or so. We go back through some of the most interesting papers and findings over the last 3 months, and we just talk about it. We try to figure out whether there's some broader theme to be gleaned from what's going on. We try to unpick what it might mean for the industry more broadly. And if you're not familiar with Veera's work, he is prolific on Twitter and on Substack on writing up stories about genome-wide association studies and other amazing genetics discoveries. So I encourage you to check it out. Veera, welcome, and thanks for taking the time to be with us.
Thank you, Patrick. Happy to be here.
So like usual, we're going to go through 4 or 5 big stories from the last 3 months. The first one is a really interesting one. It's actually an evolutionary story done out of a group that actually focuses on imputation based on rare variants and has done a lot of really great work looking at repeat expansions and other kinds of hard to sequence regions through some really clever imputation techniques. So Veera, I'll let you tee this up and why you picked it and thought it was interesting. And as always, we'll put links in the show notes. If you want to read about the papers and obviously find the link to the paper itself, then it's all there. We're going to talk about each of these for 10 or 15 minutes.
Yeah, so sure. So this is one of my favorite papers published in the last few months. And this is from one of my favorite scientists as well. You might have heard about Poro Law and Steve McCurry from Harvard Medical School. And I absolutely love all their works. It's extremely creative and, it's really super interesting work they have done in the human genetic space. And we also have discussed this before in our previous podcast episodes. I was telling you about how this evolution of, genetic discoveries using UK Biobank data, goes through we initially have genotyping data, then we have exome sequencing and then whole genome sequencing. There is a big competition to find out in all the low-hanging fruits who gets to find these findings. But at every point of time, this group of people, Poro Law's team and Steve McCall's team, they are one step ahead of time because
Yeah.
they have this, like creative way of utilizing what is already they have in their hands, like this genotyping data, common variance data, and then they combine this with all reference panel, long read sequencing panel, and then they create this imputation models to, impute rare variants even before one can, find them using exome sequencing, or impute copy number variants, or even like structural variants that can be done, only through, like long read sequencing. We have seen many examples. I think in the last episode we discussed about this variable number tandem repeats related to glaucoma, colon cancer. All of this, the common theme here is like all lot of the structural variants are basically the ones driving very well known genome wide association signals. So these are very large effect, related comparatively large effect GWAS signals that's been discovered very early in the genome, in the GWAS era. But still, we are figuring out the causal genes, causal variants. And one of the themes that I particularly like and closely follow is like the series of examples where they find this particular GWAS locus is driven by a structural variant, an insertion or a deletion or some other complex structural variant. So this comes under this, that theme. So what basically the paper is about is they have identified that a genome-wide association locus near a gene called ASIP, which has been linked to, pigmentation traits. It's very well known. It's known since 2008. First, it was reported by Decode. They found that this is actually driven by a structural variant, not any structural variant, but it's actually driven by a transposon. And that is not the most interesting part, because when they look into that, they actually found a fantastic evolutionary history behind this locus. And that's what this like makes this study really super interesting. So basically, like I said, so these Porolos team are experts in building imputation models, and they have been, identifying this findings using UK Biobank data, right? So they have found this ACAN1 for height, TMC01 for glaucoma, this VNTRs. So they came across this particular phenotype, the skin pigmentation association, ASIP, agouti signaling protein. Again, we discussed this in our previous episode on this structural variant that causes an extreme form of obesity, but that's different from what we are actually discussing right now. So their initial starting point was they see this locus and it has a very large effect size on, like skin pigmentation traits like freckling, sensitivity to skin and skin color, and also, susceptibility to cancer. And they hypothesized that it might be driven by a structural variant because there's a large effect size. One of the common features of all these kind of GWAS locus driven by structural variants have a large effect size. And that is why they started this. They looked into the long read sequencing data from the Human Genome Structural Variant Consortium. I think 36, 32 individuals that is available publicly. And they were digging into this particular locus, and they found one of the 32 individuals that were long read sequenced in the reference panel actually carried this risk allele for this ASIP, which actually is associated with increased, increased pigmentation and sensitivity, increased risk for cancer. And they found that this common variant is an LD with a structural variant. So that's the starting point. And then they digged further into this, and they found that this is actually, they initially thought it was like an insertion variant. Then on a closer look, they realized that it is actually a transposon. It's a particular type of transposon called sine VNTR ALU element. It's a relatively recent, evolutionarily recent transposon. It's a retrotransposon actually. So meaning they can actually, when they jump from one part of the genome to other, they actually leave copies of their own on the previous site. And that is also important because we will see that in the story. And so they realized it's a transposon, it's a SVA. So SVA stands for sine VNTR ALU. And they were studying this further, and then they realized that there is this risk variant that is an LD with a structural variant, which is a transposon. And when they look closer, they also saw there is another transposon, just nearby, and it is a mirror image of this transposon. And the fascinating thing is this transposon is present in all humans. It's present in all humans, it's present in Neanderthals, but it's not present in apes. So it's something of evolutionary significance. So they followed up this data, and then, like already we know from a GWAS study that this particular risk variant increases the expression of ASIP. And agouti signaling protein mechanism is that it is a paracrine hormone. It is produced in the skin cells, and it acts on the melanocyte, like say, receptor MC1R that is present in the skin, and it is actually an inverse agonist. It competes with another natural ligand of MC1R, which is, like melanocyte stimulating hormone, which is actually produced from the brain. So depending upon, if melanocyte stimulating hormone acts on the MC1R, then more, like dark pigments is produced that gives the dark pigmentation. And when agouti signaling protein interferes with the MSH binding, it actually gives the, like the light pigmentation protein, right? So we already know that this risk variant is associated with increased pigmentation, and it matches with its mechanism that if you look at the EQTL data, it increases the ASAP expression. But here they wanted to understand further how to understand this mechanism. What is the role of this structural variant, right? So how it is interfering with the expression. And they analyzed the GTEx data, and then they did, like they identified the structural variant using the whole genome sequencing data from the GTEx. And first thing they find, as was previously known, this risk variant increases the expression. So does the variant that is the structural variant in LD, that is also increases the expression. But like when they looked closely into the different transcripts, then they found there is so this way this ASIP has like it undergoes splicing to form different transcripts. There are transcripts which are coding transcripts which are form the functional protein, and then there are non-coding transcripts which are like aberrantly spliced transcripts which doesn't produce a functional protein. And so the individuals who carried this olive element actually had a decreased levels of a particular non-coding transcript, which does not produce, like a functional protein. And so when they actually looked at the, like the mechanism, like to summarize, what actually happened is that there is like two olive elements, right? So the first olive element, which is actually fixed in the human, so introduced this aberrant splicing, and that actually reduced the amount of functional ASAP produced, right? And so this actually is like probably some to like 5 many million years ago, when the hominid had split from the great apes, is just before they actually split from the Neanderthals, right? So the evolutionary significance of this is that, so at that point of time, a transposition even happened, and this oligo insertion actually decreased the ASIP signaling. This made the hominids to become more, dark. They gave it more dark pigmentation. And this helped them to survive, probably because that is the time when they were starting to lose hairs and they were getting exposed to increased UV light. So the selection pressure was intense, that only people who had this uh, transposon survived because of the advantage it gave with dark color. And this has been going for many million years. And then recently, right, so they found this polymorphic olive element, which is the one in LD with the GWAS locus. It's like maybe 25 to 30 years if age. And this is in the recent evolution and after the out of Africa migration event. And so this is actually has released whatever the disruption that reversed whatever the disruption that the ancient olive element made. And now again, the ASAP is back to its original levels, and this kind of increased the pigmentation. And so this also gave an evolutionary advantage for people who've been exposed to very low amount of sunlight, probably that gave, like helped them with the vitamin D synthesis and other stuff that is important for health. And so it appeared to be very intense pressure, because within a short span of time, this particular, this polymorphic olive element increased in allele frequency to up to 11 percentage. It is seen only in the European populations, and it is a very high allele frequency. It's almost absent in other populations. And this is the whole, like the story of how they understood what is the mechanism, like underlying this particular GWAS locus. It's really super interesting because it's also like interesting in a way that it's found in 2008, this GWAS locus. And this particular structural variant is part of the default annotation. So they say like this been known for 15 years, but no one had found that yet so
Mm.
far, right? So this kind of says that still there's so much there to be, like people are not actually noticing it. There are so much you can actually find. And I think like we will hear more similar stories. There's like hundreds, thousands of GWAS locus we have in GWAS catalog. We don't know what's most of the locus. We don't know the causal gene. We don't know the causal variant. And there is like so many such examples. When you think about it, so many such examples are actually lying there to be found. It's really, fascinating. That's why I really love the story. And this also very much in line with all the fascinating discoveries this team has made in the past using, this approach.
Yeah, it's really interesting how number one, I always love the stories that have some kind of fascinating evolutionary backdrop, that just contemplating the timescales over which these kind of things happen and learning more. We should cover this in another episode, but learning more about what ancient DNA is telling us about the dynamics between humans and Neanderthals and Denisovans, and rewriting a little bit of the history there. Anything that kind of touches that part of our evolutionary history I find interesting, but also the second point you made about some of these things are hiding in plain sight, and there wasn't proprietary data or anything like that that was brought to bear on this. It was
Yeah.
just looking at something that's been putting a couple of facts that could have been known in isolation for a long time, but just very smart people getting in and figuring out what's going on. And I do share your instinct that there's probably a lot more of this to come. We're moving into the world of Alzheimer's and frontotemporal dementia and neurodegeneration more broadly. It would be great if you could introduce this story and why you picked it.
Yeah. So this is another fantastic story. And this is, keeping our tradition of, I think like almost every episode we had a topic related to Alzheimer's. I think
Yeah.
we
Do you
continue.
think that's because this is an exciting time for Alzheimer's, or is it just we
Absolutely. I
both like
think
it?
so. Really a lot of interesting developments happening. And, I think like, yeah, I mean, we could probably, let's see how it goes, but probably we'll have this one topic related to Alzheimer's in every episode, I think. So this is slightly different from our usual, like discussions. We talk about human genetics. This is related to human genetics, but not in a way that we usually, like in the papers that we usually talk about. And so I came across this paper a couple of months ago, and this is from Huda Sokby's lab. So she's, neuroscientist, world famous neuroscientist who has worked on a lot of, like neurological diseases like Rett syndrome. And she's also the one of the board of directors at Regeneron. And I was like very interested when I came across this preprint that was tweeted by the first author, Da-Yoon Chloe Chung. So they're from the Baylor College of Medicine. And so what this paper is about is like in this paper, they describe about an isoform of tau protein that's related to Alzheimer's that is actually highly expressed in certain regions of the brain that are typically spared in the Alzheimer's, neurodegeneration. And they kind of argue that this might be an interesting therapeutic target for Alzheimer's. So I thought it was like really interesting, and then I looked into it. And so a little bit, of background of the tau protein, right? So it's actually a microtubule associated protein. It is highly expressed in the neurons, and it was discovered in the 1970s when they were like analyzing the brain tissue of pig, actually. So they found that this is so the people have been, studying microtubule. Microtubule is this kind of cytoskeleton protein, like in all the cells, in the neurons. In the neurons, particularly, it is very important for neuronal migration, but also, like communication, and also the structural integrity of the new to maintain the structural integrity of the neurons. And microtubule has been a very important topic in the early times. And when they were trying to understand how this microtubule assemble and disassemble, and that is how they found this. There is the kind of a mysterious protein that's important for the microtubule assembly, and that is when they found this protein, and they named it as tau, a microtubule associated tau protein, and that is the name of the gene MAPT. And we now know a lot about this gene. This is really important gene because mutations in MAPT itself causes a lot of early onset dementias like frontotemporal dementia, Pick's disease, like supranuclear palsy, but also biologically tau protein became like is an defining pathological feature in neurodegeneration and Alzheimer's disease and also many other conditions because of because the tau protein are very unstable protein. They can get like kind of aggregate together to form this kind of clumps, tau tangles, and which causes like damages the neurons and cause believed to cause neurodegeneration. And I think companies are right now developing drugs that target the tau protein. There are different drugs that can target the tau protein. So it's really a very important drug target. And that is why this new development is like really super interesting. And their starting point was that they came across this particular isoforms, I think. So the one of the important feature of tau protein, the MAPT, is like it undergoes extensive splicing, and there are like many isoforms are formed. It forms, and the one that we are, typically are commonly studied are the isoforms that are, very well known to be expressed in the brain. And but the isoform, which is topic of this paper, it's called big tau. It's also called PNS tau. It was discovered in the 1980s when they were studying the, peripheral nervous tissue. And so they found there is a particular isoform that includes an extra part of the gene, and then that is when they found actually there is a other exon there, and they named it exon 4A. And since then, in 1980s, it's been like 20 years, and it's always believed that this isoform is not expressed in the brain, and it is only expressed in the, like in the peripheral nervous tissues and heart. And so I think like this team coincidentally found that this isoforms expressed in the brain, and probably that what brought their interest into this. And that made, like then they found really a lot of interesting feature. So first thing what they show is actually this isoform. It's called big tau. It's expressed in the brain, and particularly in cerebellum, brain stem. And these are the regions that are typically spared from neurodegeneration, Alzheimer's disease. So they show that in both in the mice as well as in the human. So now the next question is there any link with, is there any reason, any kind of link with, like susceptibility to neurodegeneration? Because, the tau aggregation is one of the driving factor of neurodegeneration.
Just
And
sorry, quick one before
yeah,
we move on to the neurodegeneration. What changed for them to actually discover that it wasn't those parts of the brain where it wasn't known previously? Why did that previous kind of dogma, I guess, that it was not expressed in the brain get overturned?
yeah, I think like it's just people probably didn't look at it. It was not, people did not study this particular isoform in because there's like a bias towards what is commonly studied, right? So
Right.
and I think no one retested that that hypothesis that this is expressed only in the peripheral nervous tissue. And I'm not sure how this team actually came into it. I think I guess that probably they kind of coincidentally realize that this isoforms expressed in the brain because they work on a lot of projects related to neurodegeneration, right? So, and that is probably then they got more interested in this and started studying this more. And just
Sense.
because, they found some isoform in the brain that they're not supposed to find based on the literature, right?
Yeah, yeah, that makes sense.
So I think, yeah, that's probably their starting point, and they try to prove that it is expressed in the brain, and they they confirm that it's expressed in the brain using Western blot and also RNA sequencing of mice brain and human brain. And then they started studying this further using a lot of experiments to characterize it. So, like, so some of the features or characters of the tau protein in terms of aggregation is like I said, it's an unstable protein, and it has a tendency to form aggregation, right? Aggregate to form tangles. And a lot of factors actually influence the tau aggregation. One of them is the phosphorylation. So tau protein can get phosphorylated and dephosphorylated, and like that is, and the balance between the two is important in terms of they holding on to the microtubules and keeping them intact. So phosphorylation, and then like the how fast this tau protein can degrade through a process called ubiquitination, and then how much affinity they have towards, so like I said, it's bind to the microtubules and keep the structures intact. So the affinity of the tau protein with the microtubules is important. If they are not bound to the microtubules and it is in the free form, probably it start to bind to each other and start aggregating. And so these are the features that are characters that determine the aggregate, like the tau aggregation and neurodegeneration. So they tested one by one what is the character of the big tau compared to the normal isoforms that is usually studied. And they found that, like, the big tau is like less prone for hyperphosphorylation. So as people age, the tau protein get more and more phosphorylated, and increased phosphorylation is bad because it kind of increases neurodegeneration. And first thing they found is this tau, big tau is like resistant to like this increasing age associated phosphorylation. And that kind of pinpoint, hinted that probably that could be one of the factors why it is resilient to aggregation. And then they looked at the ubiquitination. So the interesting part of this extra exon that gets spliced into this big tau is there's this like lot of extra serine residues. The serine amino acid residues is actually important in the context of ubiquitination. Ubiquitination is just a process through which, so you add this kind of tax ubiquitins into this protein, and then this is like targeted towards degradation. So it turned out this big tau can be it gets more ubiquitinated and it can easily degraded, cleared off. So that could also, probably contribute or maybe explain why they don't, like aggregate to form to cause neurodegeneration. And the other feature they tested using experiment, in a functional experiment, is the microtubule binding affinity, right? So like I said, the tau protein need to be bound to the microtubule. Otherwise, they start gets aggregated, and they find this compared to the regular isoforms, the big tau actually have higher affinity to microtubules. And finally, they also actually tested the aggregation tendency itself. So, like I said, there are so many mutations in MAPT that causes new dementias, early onset dementias. All this causes this tau protein to aggregate, right? So they introduced one, like representative mutation in the mice model. And fascinatingly, even when you force the tau protein by introducing to mutation to aggregate, this particular big tau, isoform seems to be resistant to this aggregation.
Mm.
And all this kind of puts together, like a compelling story that this particular isoform is probably, more resilient towards aggregation. And so why this is like really important, because like I said, tau protein is a target, right? So right now there are different ways of inhibiting the tau protein as a treatment for Alzheimer's disease. And so the therapeutic implication here is like, instead of, completely, inhibiting the tau protein itself using siRNA or something, you can probably try some non-destructive way of treating Alzheimer's disease. And this kind of, for me, reminded of this recent Casgevy mechanism, right? So in the Casgevy, we use this kind of, we know there is this BCL11A is important for the hemoglobin switching from fetal to adult hemoglobin, right? And
Yeah.
by inhibiting the switching, we increase the fetal hemoglobin levels. So this is kind of, we are using a natural, like gene regulatory mechanism to treat the disease, right?
Yeah.
By It's the same thing here, right? So if we can find a way to nudge the MAPT protein to produce more big tau than the regular tau in the brain regions, like in the hippocampus or in the cortical regions where they are not produced as much, is it possible? Probably we can slow down the neurodegeneration, or even, like prevent neurodegeneration. It's a very interesting, question, particularly given most of the ideas so far in the Alzheimer's disease therapeutic space hasn't panned out well, right? So
Yeah.
inhibiting the amyloid beta, this whole amyloid beta hypothesis is being heavily debated. And we even saw in our last episode discussion that people who are resilient to, Alzheimer's disease with this presenilin mutation, right? So the ApoE Christchurch carriers, they do have high amyloid beta burden, but they had less tau burden. And that is what actually defined their resilience towards Alzheimer's disease. So probably the tau burden has more, you know, important in causal role in neurodegeneration than the amyloid beta burden. And if that is the case, like thinking more in terms of tau as a therapeutic target is, I think, probably will have more, positive
Yeah.
outcomes. And so in that context, I think this is really a very important finding, and I'll be like watching how this story pans out.
It's a great insight. Do you have any thoughts on how would you go about, I guess, encouraging inclusion of that extra exon to get the big tau? Are there established methods for doing that, or is that a little bit of an open question?
I mean, as a geneticist, I really want to see if there can be something like BCL11A
Yeah.
that actually controls the splicing, the isoform balance, but I don't know. Maybe we will find something like I think there's been extensive work on the EQTLs of MAPT. There's already, we know there is two major haplotypes. One of the haplotypes is actually protective. So probably, more people might study more in detail on this and can find, like genetic genes or transcription factors that might play a role. That is one way of thinking about it. But the other way would be to, like to use some kind of splice modulators to increase the splicing inclusion of this particular exon 4A to create the big isoform. And
Yeah.
that is more, like a biotech aspect, I think, to design the drug or find this particular splice modulator that can do this, probably through some high throughput screen or something. But I'm sure like people will start working on this, and because the data is very compelling in terms of the characteristic of this big tau
Yeah.
isoform.
Yeah, yeah, it's super interesting. For our third story, you shared a really interesting finding around essentially there's coming on, I guess, from the theme of groups that have a particular reputation for doing really interesting and innovative things within a particular area. Jean-Laurent Casanova's lab has a long history of doing really interesting intersections of, I guess, kind of genetics and viral infections and other kinds of infectious disease work. And this is one of a series of really interesting stories that they've put out in the last couple of years. I wonder if you could talk a little bit about this TMEFF1. I'm going to call it TMF1 for the purposes of easy pronunciation.
Yeah, so this is another super interesting story. And again, this comes from one of my favorite scientists. So I came across the scientist Jean-Laurent Casanova from Rockefeller University maybe like a couple of years ago, I think, when I came across this paper on interferon receptor mutation that they found is like very highly, like homozygous mutations, very highly prevalent in Polynesian islands and also in some other, like Greenland regions. And that was the reason for, like that actually causes increased more vaccination associated deaths because of the, like defect in the and viral antiviral mechanisms there. And so that is when I started, reading about it, and it was like really mind blown with amount of, work Casanova's team has done in the space, in the particularly the, like genetic susceptibility to infectious diseases. They are focusing more on monogenic causes. And it's really, like their team has been like working for like many years now to understand like understand the intracellular antiviral, like a lot of immune related pathways that are important for fighting immune response against viral infection, bacterial infection, also other kind of infections. And so this particular paper really was super interesting because it had a very like interesting therapeutic relevance. And so basically what they found here is that they found there are some human knockouts who lack a protein called tomoregulin, and this is expressed in the neuronal membrane. And these people are very highly susceptible to herpes simplex infection brain infection, encephalitis, herpes simplex virus 1 encephalitis. So this is a continuation of their long-standing work. So like I said, this team is interested in identifying genetic causes of, like this kind of infections, right? So they have a little bit of herpes simplex virus. So, it's the infection is very common, right? So, and usually it is, affects the nasal, lips, and oral mucosa. And this is like initially it is resolving, it resolves, and then but the virus stays dormant in the trigeminal ganglia and then like often it can have reactivation actually can cause genital herpes or other herpes. But in some extreme cases, extremely rare cases, probably 1 to 2 in 100,000 cases, this virus can get into the brain and then it affects the brain. It can go into the brainstem. It causes like hindbrain encephalitis, but also it can go into the forebrain cortex and it can cause like, forebrain encephalitis. And so this is like really, like people were interested in this because generally when we talk about, infection, immunodeficiency, we talk, it is mainly white blood cells and the pathways in that, right? But there is also one of the major, like discoveries that this team made is like there is also cell intrinsic antiviral pathways that protect specific cells from infection.
Yeah.
And one of their important cells that they were interested in the neurons. So they have worked on this for many years now. They have identified, many proteins in the interferon pathways. So interferon signaling is very important in the antiviral, antibacterial response. And then there is like proximal signaling for interferon, that is upstream and downstream. Upstream is all the pathogen nucleic acid sensing proteins. We talked about this in the context of systemic lupus erythematosus before. TLR signaling, TLR 7, 8, right? So they are all comes under the proximal upstream of interferon signaling. Then you have downstream of interferon signaling, interferon receptor. All this contributes to the like the immune pathways for the infection. So they've been identifying one by one by studying cohorts of cases who have herpes simplex encephalitis, trying to bring genetic diagnosis. And the belief is that most of these cases who get herpes simplex reactivation causing encephalitis is probably due to some genetic defect. So in this part, there is like a long series of papers they have explaining one after other gene. They came across recently this particular gene when they were studying this cohorts of cases. So most of these causes are recessive causes. So they specifically looked for recessive mutations, and they found two patients among the 331 patients cohort that they have. Most of them are undiagnosed, and they found these two patients carried a homozygous mutation in this particular gene, TMF1, encoding tauopathies. And this is very interesting because most of the findings most of the genes they find actually fall under a very well-known antiviral pathways like interferon signaling, but this stood out because nothing is known about this gene. The gene was cloned like 20 years ago, but it people know that it's highly expressed in the brain, and but they didn't know what it had a function, and that was made this like finding super exciting. So the first question is like, we know this protein, this TMF1 tomoreglin is in the neuronal membrane. It's a membrane protein. So, like obviously the hypothesis that does it has anything to do with the virus entering into a cell, right? So then they did this iPSC neurons, iPSC cells derived neurons, and they tried to see how the viral infect the cell culture with herpes simplex virus and see how the virus proliferate, grow, in the presence and absence of tomoreglin. And they were surprised to find that when you actually knock out the tomoreglin, the virus actually grows very faster. So
Mm.
obviously, this protein has some role in the virus replication. And they were trying to understand. And the initial hypothesis is that probably it is part of the cellular signaling that we know, like TLR signaling, interferon signaling. But surprisingly, it doesn't seem to have any component of this, right? And then so next day, okay, the case kind of, okay, so probably it has some kind of a role where it prevents the virus entry into the neurons. And so they were trying to see, okay, if that is the case, right? A lot of this antiviral defense, immune defense that comes in the intracellular level, right? The interferon signaling is only after the virus gets inside the cell, right? So if this protein can prevent the virus from getting inside the cells itself, so then this can actually protect even cells that are deficient in this kind of immune signaling. So they
Mm.
actually test this hypothesis using, they particularly introduce this, make these cells that are deficient of interferon receptor and they overexpress tauopathies. And even these susceptible cells, these cells that are susceptible for herpes simplex infection are protected, when you actually overexpress. That's what like, so that make this finding even more interesting because now you have like even more stronger mechanism to prevent this infection. And so they studied further, and then like we already know that herpes simplex virus requires receptors to enter the cells, and there are many receptors known. There is like glycoproteins, there are specific ligand, binding receptors, and they were like trying to see if the stromatolin interferes with any of this receptor, how actually it tried to it prevents the entry of the virus, right?
Right.
And when they were trying to do some co-immunoprecipitation experiment, they found nectin-1, one of the known receptor for herpes simplex, is what it is interacting with. So basically, it interacts with this nectin-1 receptors to prevent the entry of this herpes simplex virus into the brain, right? And the most interesting part is it's a transmembrane protein, and all you need is only the extracellular portion of the protein to prevent the infection, like prevent the virus entering. And so maybe it's maybe you can develop like synthetic versions, analogs of this protein, only the extracellular portion, and this can act like an antiviral drug, and
Yeah.
so because this protein is extremely specific to the neuron, it's expressed only in the neurons, right? So evolutionarily this has neurons has developed this mechanism. Now you found this, you can actually give a systemic protection by by exploiting this
Mm.
evolutionary. And this is kind of falls under our theme of using this kind of within individual diversity, right? So the way hemoglobin is regulated, the way tau protein
Yeah.
is regulated, and here there is a cell specific mechanism which can be repurposed for a new drug.
Yeah.
So
Could be
that's why
generalized. Yeah. Interesting.
uh, super interesting. And these findings again and again emphasize this important point, right? So traditionally, we've been like, as a human geneticist, I go look for genetic variations across individuals in the population. But there is so much we can learn from within individual variations, diversity between the cells, cell types, between the tissues. And we can actually exploit those mechanisms to develop new drugs. This is like a
Yeah.
fantastic.
Yeah, it reminds me as well as some of the interesting work going on looking at somatic variation in the liver, in the brain, in lots of other organs to not, to your point, look at variation between people, but between tissues in the same individual in this case, or even between cells in the same tissue where you have some who are in a pathological or disease state and other cells that are healthy. It expands the toolkit. And when you start to combine that with genome-wide associations and other interhuman variability, you can have some very interesting power calculations that you can do around what's possible, bringing all the different techniques to bear.
Yeah, absolutely. Yeah. So
I
much way as we have to.
Yeah, we're just getting started in a way, especially not having it's a challenge to think about how we sequence every possible tissue and disease state and time point. There's still a lot of work to do. Moving on to our second to last story, one of both of us really like the non-coding genome, and this one was a super interesting one. I'll preface this by saying when I studied developmental disorders in my PhD and was hunting for non-coding variation, this was one of the ideas that we had, that probably somewhere lurking out there would be effectively gain of function mutations that we'd find where in the in the coding region, as you pointed out in your Substack, most of what you see is loss of function. You also see lots of gain of function, but it's a lot easier to imagine in regulatory space creating a de novo enhancer, for example, that flips a gene on in a tissue that it's not supposed to be on in. And we sort of already have examples of this. Like I think about the polydactyl example is not is switching the Sonic Hedgehog gene on, and a little bit further along on the limb than it's supposed to be. But I think this is a super cool example. And I think the authors made the point in the paper, and you made the point in your Substack. This is probably the first example of anything quite to the extent that they've discovered here. I'd love if you could talk a little bit about this novel cardiac arrhythmia syndrome that's mediated by a non-coding element.
Yeah, so this is another story that falls into a recurring theme. So we've been discussing a lot of non-coding mechanisms, right? And most of the cases, it is always a rare disease, a Mendelian disease, trying to identify the causal gene mutation, and then boom, then we have like a new non-coding mechanism. We had this hexokinase disease, intronic mutation for hyperinsulinism. So, this here a new non-coding mechanism. So the starting point is like people in Copenhagen, actually they came across a series of patients who had a very specific, like a kind of a cardiac clinical feature, heart disease. So I think the index patient is a 30-year-old. It was probably, like more than like 35 years ago. And they actually found this patient. They had like a ECG pattern called ST segment depression, which is typically seen in myocardial ischemia. And it is a sign there is something very, dangerous is happening. And so probably that might have made the clinicians, aware of what is happening. And when they tested this patient, they did not find any abnormality. And they followed him for like 30 years, and he did not have any issues. So, and then after 30 years, this patient developed cardiac arrhythmias like fibrillation, atrial fibrillation, ventricular fibrillation. Then went into cardiac arrest. They resuscitated him, and they found that not only him, but also his children also had this very peculiar ECG feature. And this kind of, because they were excited about this because it was the time when Brugada syndrome, which we spoke about in our past episode, like, again, it ever story of Brugada syndrome came is similar, right? So very specific ECG feature. And then, like then they found everything fell into place about the gene. So based on that, I think that kind of inspired these clinicians to think that, okay, now probably we are, like dealing with a new cardiac syndrome. So they call this ST segment depression syndrome, and it was published in NEJM. And part of this, the authors, part of the authorship team is like the people from Oxford University, Hugh Watkins, Jim Hughes. They were the senior authors in the current preprint, and they continued working on this. They were trying to identify the genetic cause of this novel cardiac arrhythmia syndrome. And so what they did is they collected family samples. So they did this, like linkage analysis, and they found a signal in chromosome 20, and they sequenced it, and they found the haplotype. And so they, based on three different families, they triangulated the actual causal variant, and they found that the causal variant is actually a non-coding variant, and it is like a complex insertion deletion variant. So it has like a deletion of 17 nucleotides and then with an insertion of 4 new nucleotides, like they call deletion insertion TCCC variant. And so they found the causal variant, and it's a non-coding variant, and they wanted to understand the mechanism. And that is when, really things got really interesting. So they first started examining the locus in the chromosome 20. It's not that many genes in the vicinity. There was only one gene which was like 7 kb away from this non-coding variant. And one exciting initial finding they realized is that there is a GWAS locus at the same region identified for ST segment changes. There's a huge, we have like a large sample size GWAS for ECG, different component of ECG changes. They found one, and that was like made them like more, like interesting, because there is now you have like another, orthogonal finding from the GWAS. And the nearest gene is actually KCNB1. It is a potassium channel. It was not immediately evident because this protein is not expressed in the heart, and it is a potassium channel, and it is more expressed in, like in the brain. If you look at the human genetics, online Mendelian inheritance in man literature, you will see that this is actually a neurodevelopmental gene. People with, it's a haploinsufficient gene. So it doesn't make sense, like what is happening in the heart, and they're trying to understand this. and they were trying to see, okay, first they went into epigenetic annotation, all this, non-coding regulatory annotations they can, get their hands on. They found this low resolution, iPSC-derived cardiomyocytes cells, high C data, and they tried to see if there is any physical interaction. And they did find that the region of this mutation actually physically interact with the promoter of KCNB1. So that made things interesting, and they wanted to study this further. So they try to see if there are known epigenetic marks like histone marks that says that there is like enhancer or promoter elements are here. And when they looked into the annotations, they're surprised to see there is no open chromatin evidence of open chromatin region in this particular mutation site in cardiac cells, in heart cells, cardiomyocytes. But they did find that there is one open chromatin region a little farther away that is like 139 kilobase pairs from the KCNB promoter. And even that open chromatin region, which they name it E139, is not present in the, like in the heart, but it was present in the skeletal muscle. It was present in a lot of, cell lines, cancer cell lines. And so their hypothesis was, what if this mutation is probably introducing a new regulatory element, creating, increasing the expression in heart. So first thing they did was they did some kind of computational prediction by, feeding in the genetic sequence into a neural network data to see if this changes, if this genetic sequence can trigger, like a enhancer-like signature in the heart muscle. And they did find some evidence for that. And that motivated them to actually do experimental proof. They gene edited, first they gene edited the iPSC cardiomyocytes, and they did some high-C experiments, and and to do some ChIP-seq experiments, and they found there is actually when you edit the gene sequence to introduce this mutation in the iPSC cardiomyocytes, it opens up the chromatin in the cardiomyocyte. That was like the hypothesis, right? So it creates a new open chromatin. And then they went on to do the zebrafish experiment. They tagged this particular, deletion insertion mutation and also the distal, like a E139 regulatory region into a GFP protein, and they expressed in the zebrafish GFP models, and they've looked at the, like expression across the body. And as they suspected, the zebrafish that carried this insertion mutation expressed this casein, the GFP protein in the heart. And that was like really super interesting.
Yeah.
And that kind of, validated their hypothesis. And the more interesting part is like when you actually did the high Z experiment, it's in this, like in the iPSC cells, and they did not find any evidence of physical interaction between the actual deletion insertion mutation itself with the KCNB promoter. But they did find the physical interaction between this E39 and the KCNB promoter. But what they observed is that the expression of this KCNB is enhanced, right? The interaction between the E139 and promoter is enhanced in the presence of this deletion insertion mutation. And this resembles, like this kind of reminded them that probably this is not a typical enhancer, but it's like kind of like a super enhancer, which actually enhances the association between an other enhancer and promoter. And I don't think such a mechanism has been known before. And so that was like, like really interesting, because now we are learning another new non-coding mechanism through a Mendelian disease. And it just makes me wonder how much more, like wildly different mechanisms are there that we don't know, right? We discussed in the case of polydactyl, right? So the way how suboptimal transcription factor binding is like regulating the tissue specific expression.
Yeah.
And here we are looking at a mutation that introduces a new enhancer, de novo enhancer. That was like, I think it's one of the amazing discoveries in the non-coding space in the recent times. So I'm sure it will be published in some big journal.
Yeah, well, and this is a good segue maybe to our last paper, because I think it's potentially relevant to what comes next. You'll go into this in a little bit more detail, but some of your colleagues at Regeneron recently did basically a bake-off, a side-by-side comparison of whole genome sequencing versus whole exome sequencing plus a genotype array where you do imputation. And the whole genome sequence is still significantly more expensive than the whole exome sequence and the genotype array. So the question becomes, if you want to do new novel gene discovery or novel variant association discovery, how should you approach this through whole genome sequencing or whole exome plus array, given that we all have fixed budgets in this world and we can't necessarily whole genome sequence absolutely everyone? And I think one of the findings from the paper was that obviously there are more variants found from the large scale whole genome sequencing, but there were few, if any, additional associations found. And one of the reasons is because there are just so many singletons spread around across the non-coding genome, and you need kind of enough cases and controls to have any statistical power, or you need to be able to do burden tests where you combine variants together into a gene or a regulatory element. But we know so little about regulatory elements that it may not be easy to design those burden tests, and it still doesn't get you to something like this, right, where a singleton variant somewhere or a very rare variant actually creates a regulatory element that wouldn't be in any kind of burden test. So I'm interested to get your summary of the paper and also the take on what comes next.
Yeah. So I guess like we discussed this a lot of times, right? So I mentioned it like, like every time I talk about an example like this, I kind of say, I reiterate that, we can understand how this non-coding mutations act only through studying this very highly penetrant conditions, right? So where we can understand the mechanism. And, like overall, there's been a big excitation with non-coding, like genome, right? And but really, it's an ongoing debate whether we to use exome or genome. And I think we should think about in terms of common diseases and rare diseases. When it comes to common diseases, or like like applying whole genome sequencing in a population scale, like people really, it's the UK Biobank whole genome sequencing is, I think it's an experiment to actually test this, right? And so when, Regeneron actually invested a lot in one of the 8 companies that actually contributed to exome sequencing. But then there's also like a lot of drug companies invest, I think like probably 250 million or something to de-sequence whole genome sequence. Regeneron was not one of them. And so they, like us, the leadership here, they felt strongly that, such a big investment for whole genome sequencing, we could use that money to do more exome sequencing. And that is there, because they believe that more than, like going across the genome, you should actually, like increase the sample size to find more. And that is their idea. And they wanted to empirically prove this. And that's what the paper recently published in Nature Genetics from my colleagues. The first author, Sheila Gaynor, and last author, Timothy Morton, they worked on this. And so the message is that, like exome sequencing is more cost effective in terms of gene discovery. And the reason is clear. So what they basically did is like they took 150,000 individuals on the UK Biobank. We have whole genome sequencing data, we have exome sequencing data. And the traditional way we do is like we do exome sequencing, but we also have genotyping, right? So when we do GWAS studies, we use genotyping data to do imputation to cover all the common variants across the genome. Typically, we study 10 to 15 million common variants that we get, and common variant meaning like more than 1% allele frequency. Then we have exome sequencing, which gives information on the coding region. So we combine this to do a GWAS plus XWAS. And the recent technologies actually soon genotyping array will become obsolete because common variants are actually like, can be genotyped using exome sequencing itself. So you spike the region, and then you also capture all the markers that you will need to do imputation. So basically, you have like a one way to capture one shot. You capture common variants across the genome and also the rare variants across the exome region using recent exome sequencing technology. Now, whole genome sequencing, you sequence the entire genome. So what this brings extra, if you think about it, we capture common variants through this approach, through the traditional approach. We capture all rare variants from the coding regions, which is only 1 to 2% from this approach. When we capture, so what we capture from the whole genome sequencing is rare variants, but from 98% of the genome. So that kind of makes it interesting what you can find by this, such a big space in the genome, and you capture all the rare variants. And it turned out you cannot find that much. And because most of the, like the variance that we can find association with enough statistical power needs to be present in more number of individuals, common variants or even rare variants with high enough of carriers. So it turned out most of the new variants, like, like they found like 600 million variants through whole genome sequencing in the 150 individuals, but only like, probably 400 or so, 40 million, I think, through the exome sequencing. So what is the, if you look at this extra variants that you actually identify the whole genome sequencing, they're all singletons, meaning present in only one individual private variants. They really don't contribute to any genetic association. And in the rare, at least in the coding variant space, like you mentioned, that we have this burden approach, you can combine, variants together across the gene. We don't have a similar way to do it. And also we know exactly the variant, what the variant is doing, right? There is a loss of function variant, there is a missense variant. So even when you don't have enough statistical significance, when you know there is a loss of function variant, I can go and look at a handful of carriers. Okay, there are only 5 individuals who carry a loss of function variant, let's say PCSK9, and I can go and look at the cholesterol levels and then make this kind of hypothesis based, like understanding, okay, this knocks out the protein, So this kind of lookups, even this kind of lookups, you cannot do with the non-coding variants, right? I cannot just go and pick some, like some non-coding variant and say, okay, there are 5 individuals who carry non-coding variants within this region. It's because we don't know exactly. We don't have this kind of an understanding. So all this kind of really limits what you can do with the whole genome sequencing when it comes to common diseases. And if you can save this extra money to increase the sample size, you actually have a disproportionately higher yield of results. That is also shown in the paper that the authors actually downsampled the 150K to 50K and then upsampled it to 450K. So when you compare between a 50K sample to 450K sample, when you do, like genome-wide association study using exome data, exome plus genotyping data, you get like for a tenfold increase in sample size, you get like 20-fold increase in gene discovery, new associations, right? And so that is the power of sample size. So you can actually increase more genetic discoveries using exome sequencing. But I have to say this is the current state. Like things will change. We will know, like once we have more understanding of the non-coding genome, probably things, like we will have more yield from this kind of investments. For now, it is very questionable. But I'm super happy someone actually, sequenced the whole genome, because this kind of brings in a lot of new discoveries, fascinating discoveries that we can learn about, even though, like they are very expensive discoveries, right? But yeah, this is the current state. But in terms of rare disease, I don't want to like climb more on that because I'm not an expert in that. But I can see there is a general tendency to move to whole genome sequencing and exome sequencing. And there are examples which makes this case. We discussed the RNU42 small nuclear RNA gene. Makes a compelling case, right? So we missed a particular mutation, a non-coding mutation that explains 0.5% of all neurodevelopmental disorders. And this is because people did not do whole genome sequencing. And so there are examples that makes it the case. But still, if you look at the systematic evaluation of diagnostic yield between exome and whole genome in the recent times, every paper does show that there is a slight increase in the yield when you actually do whole genome sequencing. But the question is, does this increase in the yield justify, shifting to whole genome sequencing, given the cost difference? I think it's 2 to 3 times difference. And when you are really constrained with how much you can invest, if you can do exome sequencing for 500 families and do whole genome sequencing only for like maybe 150 families, I think people obviously want to do
the answer becomes obvious. Yeah, I completely agree. I'm interested to see as our understanding of the non-coding grammar and language and how it's organized improves, how much these kind of estimates can be updated. after the fact, because I just wonder if you're able to do some equivalent of a gene burden test in the non-coding region, whether you start to pick up some more interesting things. But the fact that we're not able to do it yet is just a fact. And so, when you have a limited budget of what you can spend on these kind of things, but you do have a little bit of a catch-22 here, which is when money isn't spent in the whole genomes, there's probably less data to throw into the understanding of actually what is the link between non-coding changes and disease. So I think one of the points you put in the Substack, or one of the tweet responses from Daniel MacArthur, summed it up nicely, that he was glad that somebody did it, but was glad to not be the person to have to explain this $250 million
Yeah.
to us.
I don't know if you saw this paper. Recently, there was a paper on using whole genome data to look at the association with height
Yes,
from
yeah, I did.
therapeutic theta. And it's interesting paper. They found some really, super cool findings. But look at the bigger picture, right? So they did all this kind of single variant analysis. They clumped the variants like burden approach using, like the arbitrary definitions of upstream, downstream of
Yeah.
the gene. And they found only like maybe after accounting for known genes, like 29 signals, out of the 29 signals, maybe a subset of them actually implicate any gene, like, and the known genes that they find is very few, like growth hormone releasing hormone, like some couple of gen, then they have one novel finding. I mean, 330,000 whole genomes for height, I would expect a lot more than this, right? I mean, imagine if we do an exome-wide association study when at the first time for height using 330 exomes, and I think that kind of says how much we are limited in terms of making these findings because of the limitations we talked about. How so it's a good example of how this kind of, because height is a like a kind of a reference trait we always use in terms of heritability or gene discovery and everything. So but that kind of really fit into this story that I wrote for Substack, this paper from the University of Exeter from Michael Whedon's team.
Yeah, and we spoke either earlier this year or late last year about the GWAS of height was starting to basically saturate, right? And so that also tells you something about this exomes versus genomes argument, right? Where without large scale genomes, if you're starting to saturate, then that also probably tells you there's limited gaps still to fill from the rare and ultra rare non-coding variants, right? And that may not be the case for every phenotype, but it seems like that may be the case for height. So I guess the question is that a special phenotype where it's highly, driven by common polygenic and that's an exception, or is that more the rule where we should expect that for every phenotype there's relatively little to be found? And I don't know, people smarter than us probably don't know the answer to that either. So we'll have to wait and see.
There's definitely a missing heritability gap, right? So we have 80% estimate from twin studies. Even today, there was an genetics paper, I think, that also have the sibling-based heritability estimation from the same author who led that 5 million height paper, right? Loy Kengo. And so we know like common variant heritability, and we know the full heritability of height. There is this gap, definitely rare variants contributing to this gap. And we have a paper from Regeneron. It was presented last year in ASHG. This year again, Jack is presenting. It's a XWAS of 1 million, and there they make some good argument about how much extra heritability you find from the coding regions. So ultimately we see that there is quite a big amount of heritability missing that is coming from non-coding regions, right? So, but the question is like, how many of them are large effect variants? Probably not that many. They are moderate effect, and you can find them only when you really, really have super large sample size. And so that's like, it is going to take time to have that large of a sample size for whole genome sequencing.
Yeah, definitely. We covered a lot of ground today. Is there anything that you're looking forward to in the next couple of months that you want to shout out, or are you just going to wait and see what comes out and be surprised?
Yeah, I'm working on a very fascinating paper, and I can't talk much about it, but I'm really excited about that work getting out. And it's based on like a non-European based discovery, which is kind of my favorite theme,
Yeah.
So yeah, I'm really looking forward to getting that story out sometime in the next year.
Do you think it'll be before the end of 2024, or is it going to be in 2025 sometime? Wondering which episode we can feature it in.
I'm actually going to present it in the World Congress in Psychiatric Genetics next week. Actually, we have it in Singapore, but I don't think we are pre-printing it now, but hopefully we will get the paper published in 6 months or to 1 year. So hopefully it will be one of the paper that we will discuss in the next year, first quarter or second quarter, maybe.
Amazing. Good. Well, for everyone who's not familiar, go ahead and follow Dr. Veera. He's at Dr. Veera on Dr. Spell it out fully, DoctorVeera on Twitter. And if you would like the long form version of these, or I guess probably short form compared to how long we've talked for, but the written word version, go to substack.com and search for GWAS stories. Veera, thank you again for doing this. I always have fun and I definitely learned a lot. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selective Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.